Interesting. Is this recording yet? I'll tell the story later. Anyway, okay. So I didn't want it to be the first thing on the note, talking about. Anyway, let's move on. So pigments are part of enzyme complexes that use energy from photons to catalyze endergonic reactions. Let's take a look at this. Where does that happen? The pigments aren't just free floating in the cytoplasm of a cell. Instead, we have these specific structures called chloroplasts. Well, we don't because we're not plants, but plants have these specific structures called chloroplasts. And the chloroplast, in some ways, should look pretty similar to the last organelle we looked at, which is what? The mitochondrion. It's different. It's a very different thing, but there is a similarity to it in that it is one, it has an outer membrane. All right. And it has an inner membrane, which in this case is referred to by a different name. We'll talk about that in a minute. But in this sense, yeah, and we'll talk about why it's green in a minute. But yeah, it is membrane bound, and just like the mitochondrion, which has an intermembrane space, and then an inner membrane, and then a space inside that, which we remember is called the matrix, right? We're going to talk about this as well. But this is referred to as the thylakoid membrane. But why did the mitochondrion have an outer membrane and an inner membrane? It wasn't just for show. The whole point was that it used that membrane to hold proteins, and it used the two spaces to do different things, right? Does that make sense? We're going to find a similar setup here with the chloroplast. The thylakoid membrane forms these individual discs. And they kind of look like discs. They're not really discs. They're just kind of folded structures. But when you see them under a microscope, it almost looks like a stack of pancakes or something. And we call each individual one a granum. And of course, because we know how to pluralize things then, if a single stack is a granum, what do we call a bunch of them? The grana. That's right, because gets pluralized with ah. So there we go. So an individual folded thylakoid membrane folds into these stacks called granum, a single one called a granum, called grana, and then inside of these, we find the photosystems. And again, the photosystems are the series of enzymes, I'll say protein complexes and pigments. The pigments themselves are not proteins. Pigment structure is an interesting thing in its own right. We won't get into it today, but the pigments are their own little covalent compounds, but they're held in these protein substrates, right? They're held in these protein complexes. So, as light shines into the plant, the cell membrane itself is fairly transparent. You've seen that. Even things that look colored, when you look at the cells under a microscope, they just look clear, right? Because a single cell lets light pretty much pass through it, right? Remember, obviously, because when you look at the microscope, are we lighting it from above? Right? When you looked at did you all do the onion cell in grade 10 science? When you look at the onion cell under the microscope, did you actually not? How many people have not looked at an onion cell under a microscope? Maybe we'll just do that in this course. Why not? Anyway, when you do that, the light comes from underneath, right? Light can pass through cells. But when the light hits the chloroplasts, these thylakoid membranes are full of pigments, all right, including chlorophyll, all right? So the thylakoid membrane has the photosystems integrated so they absorb light, except for green, yellow, right? And and some red and whatever, but to keep it simple. We always we're going to mostly talk as if chlorophyll does all the work. And again, the truth is that chlorophyll doesn't do all the work. There's also carotenoids and things like that. We call those antenna pigments, but we'll get to that. So does this look kind of familiar? Yeah, so these are phospholipids. I wish they didn't draw these as green in this diagram. That doesn't make a difference for you because your diagram doesn't show them as green, because it's the photosystems that have the pigments. The phospholipids themselves, this makes it look like these are like St. Patrick's Day, where like they dye the beer green, and if like you go to Boston and Chicago, they dye the river green, which seems like a terrible idea, but they actually do that. You can go to Chicago for St. Patrick's Day, and they dye the whole river green. Sorry? There you go. So anyway, there would be no reason for these phospholipids to be green. They're not. Because again, the pigments aren't it's the photosystems that contain the pigments, right? We've already said that, but it never hurts to remind ourselves of something, so I'm going to put it there. Okay, so I want to make sure we have the sense here. Some plant cells contain chloroplasts in high concentrations. Does every plant cell have chloroplasts in them? No, what would be an example of a plant cell that definitely doesn't have chloroplasts in them? Sorry? Oh, okay. Yeah, flower petals, right? They're not photosynthesizing. What else? Yeah, the sure, the stem, the stem's dead, but yeah. But like the bark, the stem, the roots, right? Where do we find the chloroplasts? In the cells of the leaves, right? Because that's what's exposed to light. So in the cells that make up the leaves of a plant, we have chloroplasts. They're a membrane-bound organelle, which means they're floating around in the cell, in the cytoplasm, but they have their own fluid inside, right? Bless you. And they are a double membrane system, just like the mitochondrion, though the arrangement is different, right? Does anyone remember what we call the folds of the mitochondrion? The cristae, right? And they have sort of that very slightly looser step, and these form these very particular looking stacks like that. So we call the inner membrane itself the what membrane? Very good, but for these, what do we call the actual membrane itself? Thylakoid membrane, good. And then the shape they form, each stack we call a granum, and all the stacks together we would say there's a bunch of grana. Very good. Okay. Do we feel like we have the geography laid out pretty well? Yeah. And then where's the green pigment itself? Where in the chloroplast? Okay, now inside the grana, or in what part of the grana? In the membrane itself, in the thylakoid membrane. So the thylakoid membrane contains all of the stuff that's going to do the doing bit that we talk about today. Does that make sense? Okay, so photosystems. This molecule here is photosynthesis. Oh my God, what's wrong with me? This molecule is the pigment chlorophyll, and you'll see there's two different versions of it that are almost perfectly identical. All right, all of that is the same. The only difference is this substituent right here. And in chlorophyll A, what is that? If you've taken chemistry, do you know what this group is called? A methyl group, good. And this would be a... Yep. A carbonyl group, right? So we're either adding a methyl group or a carbonyl group, right? So, well, carboxyl is similar, right? But it also has the alcohol group. So now, that actually is an alcohol group. I don't know why they do it as HO. But if this carbon's already making, that carbon would be making one, two. Yeah, it's a carbonyl group. Great. So this is chlorophyll. It's chlorophyll pigment, which is embedded in the photosystem proteins. The interactions between molecules and light are really fascinating and really interesting. How molecules react to light is a big thing, but basically when molecules get big enough or densely packed enough, they all interact with the light in some way. Because light is just fluctuations in the electromagnetic field, and molecules are full of electrons, and so as light goes by, the electrons in the molecules always react in some way or others. And in optics in grade 10, we learned that things can reflect light, which is really fascinating. They can refract light, and sometimes they can absorb it. Pigments are really interesting because they do both. They transmit and refract some of the light, and then some of the light they absorb. And why that happens, let me start by saying I'm no expert in that. Even if we had the time, I would have to get a textbook and sit down and reteach myself that stuff before I could really explain it to you ably. But basically, it comes down to shape, and it comes down to where the electrons end up in relation to each other. Does that make sense? And so this particular molecule, is that noteworthy? What is at the center of it? It's a big covalent compound, and then right in the middle of this segment here, what do you find? What's Mg? Yeah, so there's a magnesium. Each chlorophyll molecule has a Mg2 plus magnesium ion. At its center. This is something you'll see. Humans, for example, have a molecule that sort of famously is important to our systems. We started later on in the process that has an iron atom embedded in it. Where do we have a big complex covalent molecule that has, yeah, and what's that molecule called? That's okay. Hemoglobin, yeah, very good. I know, I can see it. I can see the wheels. It's all good. This is a thing that you see sometime where an organic compound will have an ionic component to it. It's kind of interesting. So this will naturally absorb light. And dissipate that energy. But the photosystems are responsible for collecting that energy in a usable manner. Does that make sense? We want to collect that energy in a usable manner. In other words, i.e., to build a sugar molecule, right? So how's that happen? Well, how it happens is that we need to make sure as photons come in, they collect electrons, right? And what we have is the photosystem itself is a large protein that contains not one pigment, but a whole network of pigments. So the photosystems, I'm going to say each photosystem, right? Each photosystem is a protein with many pigments which use the energy from captured photons to release Well, we've done this redox thing enough now. What do they release? It's okay. It's okay. They're going to release electrons. And if they release electrons, what they get released from, those electrons get passed along. So the electrons have been energized by the photon. They get passed along a network of pigments, all inside the photosystem. They get passed along a network of pigments. Until they reach the final one. In each photosystem, one of the chlorophyll pigments is the one that is actually designed to replace the electrons that have been getting passed around by taking the electrons from a donor and pass them along to something else. So why not just have only this one here? This is the one that's actually going to do the job. So we're going to say until they reach the reaction center, which is itself a pigment. All right, and the reaction center, and how do I show that? I'm going to say read this side first, read this side second. The reaction center pigment will use the energy to oxidize an electron donor, and pass high energy electrons onto an electron acceptor. In other words, onto the next step of the process. Oh, has it? I will find one for you. Do you need one right now? I don't have one right now. I can definitely find one for you. There may be a pair back on one of these cards back here. In fact, I know there is. Problem is my stuff's all like hardwired, like with my wires. Sorry. You know what? I used to have one here, and I took it off at some point. I should bring it back. It's nice to be able to lend that out. Okay, so guys, let me ask you. This seems all very convoluted, but if you look here, the idea is no matter which chlorophyll molecule gets the photon, electrons are going to get excited and they're going to get passed around, and they keep kind of bouncing around in a semi-random way. Though there are, I actually worked with my first year biology professor, who I had like 4 times when I was at Brock, was this amazing guy named Doug Bruce. He later became the chair of the department. He was this really entertaining guy. He was young when I was there. He's probably old now. You know, that's how time works. But he was really cool. And he, like I remember Robbie Burns. Do you know who Robbie Burns is? It's like the great Scottish poet and statesman. So you celebrate Robbie Burns Day, which I don't know if that's his birthday or the day he died. I think it's his birthday. And so Doug Bruce would come in every Robbie Burns Day wearing a full kilt and have a sword, and he would recite a Robbie Burns poem in a thick Scottish accent from the front of the room, and he'd keep teaching. But he studied plants, and he studied photosystems. I remember one day he said, guys, my research is about to come out. We've built a simulation of photosystem 2, and it's actually like molecule accurate. Sergey and I, that was his one of his postdoc students who was helping him do computer simulations, worked on this thing. Do you want to see it? And he was hyping this up, and we were all like, there's like 300 of us in this room, and this is the second year plant physiology class. And we're like, oh my God, yes. And he put this thing up, and there's this diagram like this, and it was just like little yellow dots going, and it was, I guess, tracking where the electrons were going from the photon that got absorbed. But it just looked like a bunch of flashing yellow dots on a big complex, like just like you'd see in mole view, one of those like ribbon diagrams. And we were all just kind of like, oh yeah, that looks great. And he was like so excited. He was like shaking. But the goal is to get a photon here. And if a photon gets absorbed right here, which does happen sometimes, then that's great. This chlorophyll here is designed to have a molecule donate electrons. We'll talk about what molecule it uses, but I think you can probably guess. Readily available in the cell, and then it passes that electron on to the next stage, right, to the next step. This will be like the rest of the photosynthesis process, all right? And we'll talk about that in a minute, but the basic idea is to get energy. So this is the energy coming in, and the goal is to get the energy to come in and go to the rest of the photosynthesis process. Does that make sense? That's the goal. Why not just have this one chlorophyll?
Why not just take enough? Why not just take the sugar and immediately turn it into energy?
Okay, but this does work. I will say, if every once in a while this happens to be the one that gets the photon, it will just work right away. So why not just have that one? Well, the answer is, can you catch a fish with a really small net? Well, sure, you can. What's a lot easier? Casting a wide net, right? Big net. Do you ever do this? Do you ever go like down to the pond and like catch fish with a net? I've done this with my children for, probably hundreds of hours in my life, or turtles. And like, I'll tell you, my kids have the stupid smallest net in the friggin' universe. And like, boy, you sit there all day. Greg can catch a fish really good. Claire is amazing. I can never catch a fish in the net. I can get a turtle. That's slow enough. Yeah. Well, I'm drawing it first. Then we're gonna write it. So no, it's good. You're assuming I lost track of my thought. That's a fair assumption. But here, believe it or not, catching a fish is related. This is just our wide net. Instead of having a single chlorophyll, the problem is this. If instead of this, you just had a million photosystems that each could capture a photon and do this, most of them wouldn't be doing anything most of the time. Does that make sense? So this has evolved to be a pretty efficient thing where this whole molecule contains a bunch of pigments because the photon has to hit the pigment. If the photon hits here, it's just going to sail through. Does that make sense? It has to actually hit one of the pigments. So we have a bunch of pigments, and whichever one it hits, the electrons get released and they start bouncing around until they get to the single one that is actually on the part of the protein that is designed to do the job. Does that make sense? So we call these ones antenna pigments. Pigment results in color, but a pigment is a molecule that absorbs light. In this case, we're talking about pigments like this. By the way, the other thing is, these can be different types of pigments that can absorb different wavelengths of light. Does that make sense? So you can put your carotenoids in here. So not only can you absorb light in different places, wider net, but it's also like having, I don't know, a few nets that have different sized holes. So you can catch big fish, but you can also catch small fish. Does that make sense? So this job is to capture a photon and then use the energy from that photon to steal electrons. All right? These are the electrons that actually get passed on. The electrons here are the ones that start moving, but we're going to steal their energy and actually use them to pass these electrons on. Because if we gave the actual photosystem's electrons away, what would happen? Yeah, like work, I guess each pigment could do its job once and then the molecule would be useless, right? So we pass these electrons around, but we don't ever actually give these electrons away. The electrons we actually give away, we're going to steal from a donor. Does that make sense? We will borrow an electron from this one if that's where the photon happens to hit. we'll let it bounce around until it makes its way to the reaction center. But when it gets to the reaction center, we're going to use that energy to steal some electrons from a donor, and then we're going to pass them on to the rest of the process. And now we don't just have electrons we passed on, but those electrons are full of energy. They're not low energy electrons, they're high energy electrons. And you can do a lot more work with high energy electrons than with low energy electrons, right? The exact same number of electrons go into your light bulb as come out of it. But on the way in, they're high energy, and on the way out, they are low energy. The ones that come in can do work. They're dangerous. The ones that go out, not as dangerous. Still dangerous. They can still do stuff. But anyway, different discussion. So the reaction center pigment will use the energy to oxidize an electron donor and pass high energy electrons to the rest of the photosynthetic process. In other words, this is the stage where we're not building a molecule. This stage is just for getting what? Most important thing? Energy. Then later on, we will use that energy to do stuff. So in the case of photosynthesis, the cell actually just gets the energy first. Would it have to be that way? Could you possibly design a system where the cell starts building a sugar using energy it already has and then replaces the energy later? Probably, right? But that's not how it's done here. In the case of the cell, the first thing that's going to happen is we're going to absorb energy. And the way that happens is we have these large antenna complexes, these large photosystems that have a bunch of pigments, each of which can absorb a photon, and whichever one gets the photon, the electrons are going to bounce around until they get to the reaction center. And the reaction center molecule is a chlorophyll. These are chlorophyll molecules, but there will actually also be other pigments in there. Does that make sense? This one will be a chlorophyll. Some of these will be carotenoids, some of these will be chlorophyll A, some of these will be chlorophyll B. So the photosystems act basically as an antenna for light, allowing the absorbance of different frequencies of light and casting a wider net. So we note that multiple pigments give better odds of collecting photons, right? And then different pigments in the antenna complex, can collect different wavelengths of light. So each chlorophyll, you'll remember, is better at absorbing different kinds of light, right? So if you only had chlorophyll A in the system, you're missing out on all of this light, right? Does that make sense? If all you had was chlorophyll A, then none of this would be available to you, right? That would all just be lost energy. But by introducing chlorophyll B, we cast a wider net. Does that make sense? And then by introducing the carotenoids, we cast a wider net still. Does that make sense? All right. It's just another type of thing. By the way, you know them. They have a kind of orangey color, and we find high concentrations of carotenoids in probably carrots. Yeah, carrots, right? Beta-carotene is a carotenoid, for example. Now, carrots have high concentrations of carotenoids. I'm not really sure why, because they're not collecting a lot of light growing underground. Probably just a happenstance of evolution. But they do, yeah. Yes. The photosystems are where what we call the light-dependent reactions of photosynthesis will occur. And no surprise, the light-dependent reactions, or sometimes just we call the light reactions, are the portion of photosynthesis that require light. And again, the purpose of the light reactions is to capture, and to get there. The photons don't have electrons because photon isn't an atom, but we're going to use it to capture electrons. We actually need an electron donor for that. But what are we capturing from the photon? Each photon is a discrete packet of electromagnetic radiation, and that just means it is a field, a wave that moves things in the electromagnetic field, which means each photon doesn't have mass, it doesn't have protons, neutrons, or electrons, but it does have energy. And what do we capture when we absorb it? It's energy. So the goal of the light reactions is to capture energy from photons and transfer it to electrons from a donor molecule which can be used in the dark reactions, or what we call the light-independent reactions of photosynthesis. We're just going to say for now, be used to do work. So this is yet another case of those redox reactions, right? Where we're going to do a reduction reaction. Some poor donor is going to lose its electrons. Is it oxidized or reduced? It's oxidized. It's going to lose them first to the chlorophyll molecule in the photosystem at the photocenter there. So while that donor is being oxidized, that photosystem is being reduced, right? And then that photosystem is going to pass those electrons on, at which point the photosystem will be the thing that is oxidized, and the thing it passes them on to will be reduced. And eventually, where are those electrons going to end up? What's the last thing that's going to get reduced in this process? What does photosynthesis make? Sugars. So in the end, we know that this is going to be a series of redox reactions, right? We know that it's just going to be a series of electrons getting passed on. The thing that stores the energy is oxidized. The thing that releases the energy is oxidized. The thing that stores the energy is reduced. And we know that because for it to make all those bonds, right, it's going to need energy. It takes energy to make a bond. It takes energy to hold something together. Yeah. Yeah. So that molecule there is what a single chlorophyll pigment looks like. And I guess I'll try to show you the photosystem. The photosystems are big. This is a big protein complex. Because again, the photosystem doesn't have one of these in it, it has many. And it has a bunch of chlorophyll A, it has a bunch of chlorophyll B, and it has a bunch of carotenoids. So let's see, where's MolView? I don't want to show you my grades. Okay, moleview.org. MolView might not actually be the best way to look at the photosystems, but that's okay, let's try it. Oh, there we go. Okay, we'll look at photosystem 2. Okay, so photosystem 2, she's a big old protein. But look, do you see inside that big old protein? I've got my computer in battery saver mode, and she's feeling it. But do you see all those things that aren't protein? Note, the purple is protein. That's the ribbon structure. Actually, because we get the lights, because this is hard to see if it's not. By the way, the purple kind of spirally ribbons. What secondary structure is that? Okay, on my computer they're kind of purpley, so just be quiet. That's really not the important part. Alpha helices and these flat blue cyan. Do we like cyan? They just look kind of washed out on yours. That's because I have this projector set up for brightness, and when you go bright, you lose color. Anyway, those are the beta sheets. And then we can see these white string bits. All of those are amino acid structure. And I wonder if I can actually briefly make those disappear. Oh, that's going to be worse, not better. Actually, you know what? That's not better or not worse. Sometimes they show the alpha helices just as these kind of cylinders to simplify. All of this, these are all pigments. So a single photosystem, do you see how just jammed full, there's just pigments embedded all throughout it. So the photosystem is designed to hold just a ton, like a metric ton of pigments, and any one of those, if a photon hits it, it will absorb that light and it will bump electrons around, and they'll bounce around, bounce around, bounce around, bounce around until they happen to get to the one that is bound to the active site of the protein. Does that make sense? Yeah. It doesn't. How to describe this? Like, because, so the answer to the question, why do pigments absorb light, gets into actually a lot of quantum chemistry, because it really does have to do with the interactions between the waveform equations of a wave of light coming in and the waveform equation of an electron. We didn't even get into that even in grade 12 chem. If you take first year university chem, what you're going to learn is that what we describe as an electron in orbit is really electron's wave equation, which basically means just like light behaves like a particle, electrons actually behave like a wave. They can be modeled by a wave. And basically, if you take all the waveforms of all the electrons in a molecule and have it interact with all the waveforms of a beam of light coming in, the math says that in certain positions of electron arrangements, a waveform of light of a certain frequency will get absorbed, but others won't interact at all and they'll just pass right through. That's really complex math. I studied it a long time ago and probably semi-understood it, but could do the math then, and it's been a long time since. So I'm speaking here from a position of being familiar with the fact that that's some math that a person could do. But if you asked me to do it, if you gave me a second year quantum textbook and said, do this math right now, I would have to do some studying before I could do it. But the short answer is that light is an electromagnetic wave, and electrons are also an electromagnetic wave that also has a mass property, and they interact with each other. And depending on where you put those things in three-dimensional space, some of them will interact, and some of them will leave each other alone, and some of them will interact by absorbing, and you can use math to predict that. Having said that, I don't know that we have done so for a molecule as big and complex as chlorophyll A. I don't even know if, when I was in school, I don't think we had an exact model of why that absorbed light. Maybe now they do with more advanced computing, but that would be a very complex set of equations because, again, if we look just at chlorophyll, which is the bit that's actually absorbing the light, so we say chlorophyll A, that's a big molecule. It's not nearly as big as, as the photosystem itself, but you can still imagine there's a lot of electrons there. Don't trust that shape, interestingly, because it's showing all of this as perfectly flat, even though those are all... Oh, see, there must be a double bond on there. Oh, there's a double bond. Why don't these have a double bond showing? Yeah, see, so this shouldn't be perfectly flat. I don't trust this model right now. That's interesting. Because for those of you who took organic chem, you should know that these are sp3 hybridized, so they should be bounced around. So don't trust the geometry of this. I'm not sure why it's showing up that way. Probably because I changed a setting in here and it's probably still in protein mode. And in protein mode, it treats all of these molecules as flat just to save processing power. But anyway, the important thing is, yeah, basically light strikes here and interacts with the electrons, yada, yada, yada. Some light can get absorbed, some can't. Did that remotely answer your question? Good. Yeah. Yeah. Mostly I say there's no such thing. Yep.
And it might just be because of wavelengths or whatever, that's not even your answer. But if green is reflected, why is blue? Blue and yellow make green.
Okay, blue and yellow, that's a great question. Can I tell you, blue and yellow don't make green. It's actually a lie that when you shine blue light and yellow light together, it makes green. When you shine blue light together and yellow light together, we see green. But that's only because our eyes don't actually see color. Our eyes interpret color based on a very limited, we have a very actually, even though it's incredibly complex, in terms of compared to what color is, we have a very unsophisticated system. There's a whole rainbow of light, and our eyes have three sensors, but our sensors aren't designed to see one specific color. They see kind of a broad range. And when we actually don't have a sensor that just sees red light. We have a sensor that sees from the red into the oranges and yellows. And then we have another sensor that sees yellow, but it sees some of the oranges and it sees some of the greens. And then we have one that sees blue, but also sees some of the greens and the violets. And the way our eye interprets color is by looking at the ratio of how those three sensors are spiking, right? But that's actually a guess. So there is nothing happening with waves when you shine a blue light and a yellow light together that make green light, which, by the way, don't. That's pigment. What would you shine together to make green light? Nothing. You have to use green as one of the primary colors of light. It's RGB. Do you know how you make yellow? Yeah, but do you know what you shine together to make yellow? If you think about fooling our eye, so if we think about like cone cells, conch cells, cone cells, and RGB spectrum. So when you do it with light, these are the three cone cells we have, and you can see that two of them actually occupy a really similar range. So what we call our red sensor sees very similarly to what our green sensor sees. But if you want to make yellow light, quote unquote, make yellow light, you will trick the eye if you shine green light and red light together, because your eye can't tell the difference between real yellow light and a combination of green and red light, but there is a difference. And you can prove it because if you shine a real yellow light on a that's a bad example. But if you shine like a real yellow light on something, that has a pigment that absorbs red, it won't actually absorb any of that yellow light, and it'll reflect back yellow light to you. But if you shine like my RGB LED lights yellow light on that, it will actually reflect green light back on you, because real yellow light is just real yellow light. But we can trick our eye by making fake yellow out of red and green. And again, a species that had more evolved eyes than us would see the difference and it would look fake. Have you ever seen the early days of color film when they used the two-strip Technicolor process? So the two-strip film process. Modern, it occurs to me that could, let's try, how do I, okay, two-color film early technology. In the earliest days of color film, they didn't have the technology to get red, green, and blue, so they shot things with only two colors. And if you watch those old color movies, everything kind of looks ridiculous and wrong color-wise because it just didn't have enough colors to trick our eye. And it got kind of close and it didn't look very real. By the time they realized that since we have three color sensors, if we put three strips of film together, and in the early days, they didn't know how to put three colors all on one piece of film. So color movies shot from like the 40s to the late 50s, the camera actually had to run three strips of film next to each other at the same time, if you can imagine. And it put a red filter in front of one, a green filter in front of one, and a blue filter in front of the other. And they had to record those. And then when they projected them, they had to project all three and line them up. Then later they learned to make a single piece of film by taking those three and using them to dye a piece of film. And then only later on did we learn how to put an emulsion where we put all three on there. But that's only because it can trick our eye. Combining different colored lights in real life doesn't change the color. They're just both there. Does that make sense? In real life, when you shine red light and green light on something, red light and green light bounce off of it. It's still just red light and green light. Your eye sees it as yellow because your eye can't tell the difference between that and real yellow light, but it doesn't magically turn it into yellow light. There's nothing happening to the waveforms that makes that happen. And if you had to shine yellow light on a certain special kind of plant that grew in yellow light, and you did it by combining red and green light, the thing would die because it wouldn't actually be getting yellow light. Your eye would see it as getting yellow light, but really your eye would be tricked. Just like this screen right now, at no point is it white. Do you have it? Anyone who wants to can take your cell phone out. Does anyone have a cell phone where you can set the shutter speed on your camera? Does anyone have like manual settings? Set your shutter speed really high and take a look at this screen and see what you see. Actually, I have a camera here that does that. There's my camera. There's my camera. Oh gosh, why can't I find where that goes in? Okay, so I actually have the technology here. Look, ready? Um, camera. There's my camera. Oh, look at you guys. So I'm gonna set the ISO to auto, and so there you guys are. I'm gonna set. If I look at the screen here, you can see I can see my screen, right? And you can see that some of it is. Can I full screen this? Oh, which one is it? That one. Okay, so we can see I can see the screen up there, right? If I turn my shutter speed up so that it takes a picture for less and less time, you're going to see something interesting happen, which is that the screen, I need to make the screen have color on it. Let's stop being full screen. Right? Look at the screen. What's it actually doing at a given time? At any given time, it's actually only one color. And this is throwing the white balance in my camera off, but if I could sync myself up with that, I could get it to actually show only one color at once. If I could find the exact value of its shutter. But the screen tricks your eye into thinking it's seeing colors when really it's only ever having a fully red screenshot at you, or a fully green screenshot at you, or a fully blue screenshot at you. And your eye just can't tell the difference. Oh, look at y'all. Smile. Okay.
Ew.
Who said ooh?
Me.
I'm talking about my friend that way. All right, guys. For those of you I don't teach another class, we'll see you Monday. Have a wonderful day. Be kind to one another. Don't do anything Toby wouldn't do. Lunch, right? See you then. Will you do me a favor? How would you feel about running down with 5 minutes to go, and when they make the cafeteria announcement, have Aiden, in a very excited manner, also mention that there's a film club meeting in room 80?
Alright.
It's dark in here.
Right? Ah. Oh, there you are. Look at you.
What is this?
Oh.
Yo, you streaming? Yo!
Oh, I guess I
Like
am.
it.